Yes, sir.
Yes, sir. Yes, sir.
Good morning, everyone.
Very good morning, sir.
Good
Thank
morning,
you.
sir.
Thank you. Once again, we are here for this live free webinar. We thank you again, Susri, for wonderful introductions. First and foremost, I extend a heartfelt welcome to all these attendees of today's distinct webinar. I am Dr. Gopal Krishnan Purohit, the CEO and co-founder of Heredity Biosciences and your convener of today's events. It's a pleasure to have each and everyone with us today's webinar. In today's webinar, we investigate the dynamic field of meta-nanoparticle synthesis, characterizations, and its applications as anti-parasitic and antimicrobial agents. As you know, metal nanoparticles are revolutionizing medicines with their precise antimicrobial and antiparasitic properties, offering innovative solutions to combat infections while addressing challenges of resistance through advanced synthesis and characterization techniques. So we are honored to have Dr. Binod Kumar as our guest speaker today. Dr. Kumar served as an assistant professor at Sri Vishnu College of Pharmacy, Bhimavaram, Andhra Pradesh. Sir, your expertise has significantly enhanced our webinar. The entire team of Heredity Biosciences greatly appreciated your willingness to share your knowledge with us. Thank you, sir, for your time and insightful presentations. We really apologize for any delays leading with the webinar and truly value your presence. Today's inspirational quote of the day: unlocking the potential of metal nanoparticles, bridging science and medicines to combat parasites and microbes with precision, innovations, and sustainable solutions. So now the highlight of this topic, our guest of speakers, so Dr. Binod Kumar will be elaborating to these presentations briefly, but a little bit highlight to the session of the topic. So coming to these introductions to metal nanoparticles, what makes them unique and indispensable in modern science? So what are the unique properties of metal nanoparticles is that it is depend upon the size of the size dependent properties. It enhances the surface to the volume ratio, and it altered metal points, melting points, mechanical strength, and electrical conductivities. When we discuss the optical properties, that is localizing a surface plasmon resistance, lead to unique color and light absorption features. When we discuss the catalytic activities of these particles, that is accelerate chemical reactions due to the high surface energy. But why metal nanoparticles is a matter? It is matter up to the medical applications. So that is targeted drug delivery systems, diagnosis therapeutics, and imaging and therapeutic agents to use it. If we will use for this environmental impact that used for the water purifications and pollution degradations also. Antimicrobial and anti-parasitic rules is effective for this broader range for that effective agents of multi-drug resistance pathogen and parasitic organisms. How to synthesize metal nanoparticles? That is three-way primary way to approach to this synthesized of metal nanoparticles. That is chemical methods, physical methods, and biological methods. If it is a chemical method, that is reducing of metal salt using reducing agents like sodium borohydrate or citric acid. It is a poor precipitations, that is simultaneous precipitations of metal ion under controlled conditions. If it is a physical method to synthesize, that is laser abrulations, generating nanoparticles with laser balls on metal targets, and evaporation condensations, that is vaporizing metals and condensing to the nanoparticles. Biological method is very simple. So this green synthesis of nanoparticle that plant extract, bacteria, fungi that used for this stabilizing agents. That one of the example is to that silver nanoparticle to synthesize using for this neem extract. Some of the factors is affecting that synthesizing of that pH, temperature, metal salt concentration, and choice of reducing and keeping agents also use it. Now, characterization of metal nanoparticles, some of the various way to characterize it, that is UV visible spectrophotometry, dynamic light scattering, and X-ray depreciation XRD, TEM also, transmission electron microscopy, and FTIR study to use to characterize to the metal nanoparticles. One of the most interesting and advanced thing is that future perspective. What are the future perspective and challenges? That is, one of the opportunities to developing novel hybrid nanoparticles for targeting therapies and using drug delivery system for precision medicines. What are the challenges we are facing for that? That is toxicity and environmental impact, one of the major challenges. And second thing is that high productions and cost and scalability issues also facing for that. Once again, I extend a heartfelt thanks to our guest of speaker, Dr. Vinod Kumar, for enriching our webinar with your expertise and insight. Your contributions are immensely valued, and we truly appreciated your times and knowledge. Now, attendees, before we begin our session and introduce our esteemed guest, let me share a brief overview of heredity biosciences. Really, I am filled with immense pride and gratitude to introduce heredity biosciences, a dream that began with a vision and the innovations and collaboration and contribute meaningfully to life sciences and beyond. Established in 2018 and rebounded its 2021, Heredity Biosciences has emerged as a trusted pillar in the medical and scientific community through the tireless effort of the passionate and diverse team, that is, the scientists, professors, and healthcare professionals who have shaped the institutions that thrive in excellence and integrity. Our team expertise in genomics, proteomics, metabolomics, and bioinformatics, and many more. Together, we channel our knowledge to provide solutions that not only for the address current challenges but also redefine the future of life sciences. We equipped with the latest equipments inside of this lab, and we support the hospitals, life science sectors, pharmaceutical companies, and we are focusing to those innovations that touch the lives and providing for the different type of services. Our missions does not stop to the delivering services. We aspire to the empower individuals through molecular biology training, analytical services research, and life sciences program into the industry needs. We are privileged to have a strong scientific advisory board also. They are timely guide us to how to improvise to our quality, cost, effectivity, and impactful outcomes remain to start every day they will be helping us. In collaboration with Heric Life Sciences, so they are not just building out the partnerships, we are creating the legacy of progress. Together we struggle to redefine the boundaries of research and healthcare, and such type of webinar we are conducting, they are helping us. We are established in Mayfair Lagoon Road, Jayadepur, Bhubaneswar, Odisha. Now, the service of this offer to this heredity biosciences, it is a personalized to support and advance research across various scientific disciplines. Our offering include analytical services that techniques such as LCMS, LCHRMS, GCMS, and FTI to deliver precise and reliable results for pharmaceuticals, environmental and research laboratories, and complex biological studies. In the area of phytochemical services, we specialize in the extractions, identifications, and quantifications of plant compounds, aiding in drug delivery systems, nutraceutical development, and many more. Our microbiological solutions include pathogen identifications, antimicrobial assay, and environmental sample analysis, ensuring safety and quality across research industries. We are providing metagenomic analysis also, and including microbial genomic sequencing, target and amplicon sequencing to explore the microbial community to diverse environments. our oligonucleotide synthesis, and Sanger sequencing. That is wide services right now and cater to the various genomic research need to the high quality, valuable oligos and reliable sequencing results. Additionally, we have four whole genome sequencing, species identifications, qPCR services, gene expression analysis, and proteomics analysis also. All these design empower research with comprehensive data insight into this research. At Heredity Biosciences, we are committed to delivering precise and innovative solutions that drive the scientific discovery and innovations across the multiple fields. Now, one of the another portfolio that is Heredity Bio Academy. Heredity Bio Academy offers hands-on training programs ranging from basic to the advanced levels. So this is the students and professionals in the life sciences sectors. Our curriculums includes diverse disciplines like clinical microbiology, molecular biology, recombinant DNA technology, and advanced modules that is advanced microbiology, molecular cloning, recombinant DNA technology, histopathology, proteomics, specialized modules that is real-time PCR, clinical embryology, and other modules also. So we have not participate with the practical experience, all the students and the essential theoretical knowledge and academic professionals given for that. We are guided by the experienced scientists and educators, training engaged in the laboratory techniques, data analysis, how to do the research, how to do the research methodologies instead the robot learning experience. So our program are designed to bridge the gap between academic concept and real-time applications, empowering individuals to excel the perspective and the field of life sciences. The students have some of the highlight of this heredity biosciences that is a broader range of comprehensive modules and this program, the training program to these BSc, MSc, MTech and other life sciences students covering to the molecular biology, microbiology, nanotechnology and bioinformatics also. And we have a hands-on experience and practical exposure to advanced techniques like DNA, RNA extractions, PCR, real-time PCR, microbial culture, and nanoparticle synthesis. So many things are given for that according to the modules of complete hands-on experience. Our expert mentorships guided to the experienced scientists, professors, and industry professionals to ensure in-depth learning of the students so that they will be excel to the experimental things. Next, industrial ready to skills. All these modules are prepared to that the industry-based standards to enhance the employability in the research, diagnosis, and the pharmaceutical sectors. Finally, we will learn about the certificates and placement supports also, and we are recognize the certifications upon completion and 100% placement assistance to all the students to try leading the companies and the research institutions. So, Heredity Biosciences already announced for the hands-on master thesis dissertations program projects in a life sciences students. Already, so many students enrolled to this dissertations program. If anyone to interested to join for this dissertation program to this our institution, our program is designed to the equipped scientists as well as the renowned educations to practical experience to the students in the area of whatever you interest. So, Heredity Bioscience proudly announced that last 3 months, more than 500 students have successfully completed the free training program. So, that is a bioinstrumentation, nanotechnology, biochemistry programs also that is a completely free program. This achievement reflect our unwavering commitment to the nourishing to the next generation of scientists and empowering them to the essential knowledge of the skills so that the students at least know the laboratory skills and instrumental skills so that will be utilized to this for the future career. These are the pasted photos are there in the slides. So this is the witness of this completion of the programs and the students will be achieved to these things and we are providing for the certificate on these free programs also. On December 8, 2024, this is the day we are really proud to that in Heredity Biosciences. We are celebrating and we are sharing to these milestones what we are did for that. So this is the day we are celebrating to the Foundation Day in 2024 at Heredity Biosciences. I'm really excited to this day, and we are. Our team members as well as for these intern students both are celebrated in this Foundation Day with a greatness. This day, future employees, recognitions, awards, students' completions in other art, drawing, and quiz, and captivity muscle performance. I'm really stunning to that. My intern students, they are well. Doing for these other art competitions, drawing as a marvelous drawing art to that, and finally we are given for this prizes awarded to this all this some the interns, and with the joy of the solidarity among the employees also, they are doing for this best work to this year. So we are given for this award ceremony to the journey of this our team members, and the celebration mark our journey to the growth, innovation, dedication, excellence of the life sciences. And all these members are doing for this best effort to these things. So in this day we are celebrated. It's proudly we are doing for that. So this slide is a very recent activities, placement activities is going on to that. Already 100 plus of the students are already placed into that, and we are proudly announced for that the successful completed for these 100 plus students into this esteemed organizations. They are studying for that. Their internship in our laboratory and joining for the different industries and healthcare sectors also. The remarkable achievement highlight to the academics commitment to excellence in the biosciences education and skill development, and congratulations all the students to securing the position to align with their aspirations. Some of the students also already have joined, and they are waiting for that the full letter, and I proudly as a mentor, proudly feel to that my students also join in different organizations. I extend my extend gratitude to the placement teams and partners, organizations of their unwavering support and their bright future. Bright future. So congratulations all these students. One of the greatest and things we are doing inside of this Heredity Biosciences, that is that announcing a collaborative partnerships with a Dhinkanad Autonomous College and Saidabad Autonomous College. These are the two institutions right now. We are MOA Science, which is their commitment to fostering innovation, advancing biosciences education, and promoting research excellence. So this day we extend our heartfelt thanks to both of the institutions to joining us in the journey of empowering students, facilitating knowledge, exchange, and driving impactful scientific progress. So we are trying to do all these activities inside of these organizations, so empowering to the students of experimental knowledge to the students. So we try to create opportunity, also creating to the frontline research and the professional developments and hands-on experiment. We're going to these institutions and doing for them some of the activities, so that the students will be empowering to the research. And thank you for the trusted and collaborations both of the institutions. So Heredity Biosciences always doing for this all this activity work in our institutions. Now that's one of the greatest achievement of this last month. So our team members and the team members, they are going for this attend the national conference and of the team. They are achieved for the remarkable success for the national conference of Saidabad Women's University, and I'm really proud to this both of the members, and they are both talented members. They are attending to the poster presentations award, and you don't know that we are stunning to that. Our institutions getting for the first prize of this Swagatika, and secured the first positions, and the Mr. Siram Behra also prized for the third prize of the poster presentations. All the out of the 50 participants, we are getting for this prize, and this outstanding performance reflect to the dedication, hard work, and excellence in the fields. And this is the big thanks for all these team members and mentors to the support of this effort. So keep signing at achievements of the new site. For the future, your career should be bright to keep going on to the same thing is to the for the future also. Last week we are doing for this. We are conducting 2 days workshop at CB Raman Global University, and this is successfully conducted to 2 days workshop also, and that fostering knowledge and hands-on express complete hands-on knowledge into the front of bioscience research. That is a microbiology, bioinformatics, and molecular biology experiments. We did inside of that. So these are the pictures we're doing for these experimental things. The students are really pleased to all these experimental things, and the event featured the engaging the sessions, practical demonstrations, and whole day and interacting discussions involving participants with available skills and insight into the advanced biotechnology and its applications. These are the pictures, and the students, they are really pleased to all these experimental activities also, and finally we were given awarded for the certificates all these students. In last couple of days, couple of weeks, the 3 publications we have inside of this heritage biosciences, and my team members, they are proudly to published to 3 papers published out of 50 publications right now we have, and in this couple of weeks we have published in the 3 papers, and this is the Sri Swagatika Subadarshini also is the first author of these things, 2 papers, and the third one is the 4th authors, and 200 plus NCBI sequence omissions and 3 patents we have right now, and we have a recent journeys, recent experimental journeys going on, and some of the research papers we are already writing for that in the student perspective as well as the inside of this experimental research perspective. For the future, we will try to write to the more publications to the future. Now, audience. Now, we are honored to welcome Dr. Vinod Kumar sir as our esteemed guest speaker. He is a dedicated professional with improved academic and professional path in the field of biotechnology and nanotechnology. Now we are introducing for that. His remarkable journey is a testament to his passion and advancing science and contributing to innovative research. His academic journey, he held in a PhD in pharmaceutical biotechnology from the National Institute of Pharmaceutical Education and Research, Hajipur, where he conducted frontline research from 2017 to 2024. He also completed his master's in pharma biotechnology from Naipur, Guwahati. Additionally, he earned his B Pharm from his Venkateswar College of Pharmacy in Srikakulam. He has a rich professional history, including his current role as an assistant professor at Sri Vishnu College of Pharmacy, Bhimavaram. He has previously served as a trainee scientist at Bhimta Lab Limited, Hyderabad, at the BCL training in the R&D Bios Development at Intas Pharmaceuticals, Ahmedabad. Furthermore, his tenure as a junior research fellow at IIT Guwahati solidified his expertise in advanced research methodologies. His current research investigates into the synthesis and characterization of nanoparticles and their applications of antimicrobial and antiparasitic therapies. His work explored therapeutic interventions targeting oxidative stress, ER stress, inflammation, and mechanism of apoptosis, aiming to decode their effect on various pathological decades. His academic journey is decodes with notable achievements, including qualifying prestigious examinations like GP80, GATT, and NIPER with an All India rank of 14 in 2017. His research contributions include publications in esteemed journals such as SAS Microlator and Frontier in Bioengineering and Biotechnology. In addition to his academic honors, he is a certified Android developer and possesses a diverse skill set, including expertise in bioassay, cell culture, and advanced pharmaceutical techniques. His dedication to science and education is truly inspiring. His achievements highlights his commitment to fostering innovation and collaborations in the domain of biotechnology and nanotechnology. We look forward to his valuable insight and contributions during this event. Today we have a privilege to hosting Dr. Vinod Kumar, who will share his expertise in a webinar entitled Metal Nanoparticle Synthesis, Characterization, and his application as an anti-parasitic and antimicrobial agents. We are excited to learn from him and engage in a fruitful discussions now. Now, sir, I request start your presentations. So these are my team members, incredible team members. They are truly work hard to that, and I'm really grateful to these team members are present here with me. Thank you everyone to listening.
Thank you, sir.
Sir, you start your things now. The
Yes, sir, if you can make me a presenter, I can share the slides, sir.
Okay.
Good morning, my dear audience, and thank you, sir. Thank you so much for the kind introduction by Susira Madam and Gopal Purohit, sir. Good morning, everyone. It is an honor to be here with all of you today, in the most important day, Foundation Day, to be part of. Honored. Thank you for giving this opportunity. And now we are going to discuss about the topic on synthesis. Sir, am I audible, sir? Hello?
Yes, sir. Yes, sir.
Okay.
Yes, sir.
So today we are going to discuss about the topic on synthesis, characterization, and mechanistic studies of allantoin, synthesized silver nanoparticle against parasites and fungus. Myself, Vinod Kumar Razana. Yeah. At first, the concept was introduced by famous professor, Dr. Richard Feynman, and he discovered the fullerene, which leads to the emergence of nanotechnology field. And the nanotechnology was the term coined by Norio Taniguchi in 1974. It is a study of manipulating matters in the nanoscale, and the advantage, as it was mentioned by the Gopal Purohit sir, it having a high surface area, which can give rise for the increased reactivity and binding potential. And along with the high surface area, it is also having unique optical properties, exhibits fluorescence and SPR properties. The nanoparticle technology has a wide area of applications like energy, medicine and drugs, and nanobiotechnology, nanodevices, optical engineering, and every field like bioengineering, cosmetics, and nanofabrics. When it comes to the statistics, from 1996 to 2000, the metal nanoparticle research and FDA-approved nanomedicines are more. And now the clinical trials are also even high in metal nanoparticle research. And most drugs are against cancer, but silver nanoparticle or metal nanoparticle against basic drugs against fungal and parasites are very less. So there is a scope for the research in this field. So I started to synthesize the nanoparticle against Leishmania disease, which is a parasitic disease. So Leishmania is one of the most serious topical infectious disease caused by the protozoa of the genus Leishmaniasis. And the Leishmania parasite undergoes two major morphological changes between the sandfly and the mammalian host. The flagellated promastogotes residues in the gut of the sandfly. Sandfly is a host, and non-motile amastogotes inhabits the SD phagolysosomes within the macrophages in the mammalian host. So it is the second largest infectious disease after malaria caused by diagentic parasite, and it infects mononuclear phagocytes like spleen, liver, and bone marrow, and it causes the enlargement of the spleen, liver, and bone marrows. And it's more prevalent in India, Bangladesh, Nepal, Sudan, and Brazil. And in many places of Bihar also it is more. So I did this research in Naipar Hazipur, which is in Bihar. So it is the local disease. As per the WHO, a total of 0.7 to 1 million Leishmaniasis cases are estimated with an alarming rate over a year. Two species which gives rise to the visceral Leishmaniasis form are the Leishmania donovani and Leishmania infantum. And based upon the transmission characteristics, visceral Leishmaniasis can be divided in two types. That's like zoonotic visceral Leishmaniasis, which transmits from animal to vector and to the human, and anthropogenic visceral Leishmaniasis, which transmitted from human to vector to humans. So incubation period for this disease is 2 to 6 months. The presently ongoing drugs are sodium stibogluconate, amphotericin B, ambizome, miltefosine, parmomycin, and pentamidine. Every drug has its resistance and toxicity, so there is a need for the natural compound with more safety, like a allantoin. So I proposed this allantoin drug in the nano formulation, and it is the other names of the allantoin is glyoxyl diurate and allantan. And most mammals process purines to allantoin, but however in higher apes and humans, the urate oxidase enzyme is absent, so they convert this into purines into uric acid. This allantoin present most in the comfrey plants and urine of the most mammals. And in the blood of the humans, like 7.2 to 48.2 millimole per liter. And its pharmacological activities like wound healing and tissue regeneration activity, and pain reducing and gastroprotective effect. And I want to make it in a silver nano formulation. So a little introduction about the silver nanoparticles. The nanoparticles are used like silver and gold are mainly used and modified to enable their uses as diagnostic and therapeutic agents. And presently, Samsung has created and marketed a material called silver nano, which includes silver nanoparticles. The available silver nanoparticle products are like Acticoat, Bactigras, and Aquacel are example of silver-based biocomposites approved by the US FDA for wound dressing applications. These are some of the allantoin nanoformulations, like a review literature, which what they have done earlier, like despite having multilateral pharmacological activities, they have made antifungal activity of copoba oil with solid lipid nanoparticle besides incorporation of allantoin in the formulation. And they also made biomedical applications with nanocomposite by using allantoin loaded porous silica nanoparticles. And to investigate the performance of conventional as well as new argan oil enriched liposomal containing allantoin as the dermal drug delivery systems are investigated. And the chitosan membrane, allantoin incorporated chitosan membrane also evaluated for its wound healing and tissue regeneration properties, as this supports that the allantoin has some pharmacological activities. So we made the allantoin silver nanoparticle as the allantoin having hydroxyl groups which can donate the electrons to the silver, and then the silver 2 plus convert into silver atom, silver Ag0. So allantoin can reduce by donating the electron to the silver nitrate, and it forms the silver nanoparticle. and that nanoparticle color will be appeared. At the starting stage, it is uncolored, the solution is, but after 90 minutes, the solution will become dark brown, which shows that the nanoparticle is formed. But after the nanoparticle was formed, as mentioned by our Gopal Purohit sir, we need to go for a characterization. I did the UV visible spectroscopic characterization, and I found the peak at 450 nanometers. And the 450 nanometers peak is significant for the silver nanoparticle. And in the FTIR data, it will we check the FTIR for the presence of a allantoin in our nanoformulation, and in the FTIR data, the fingerprint regions, that's like 1400 to 400, was matched. It shows that in the formulation, the allantoin is present. And later on, I did for size, stability, and geotopotential studies by dynamic light scattering instrument, and I found the size is 128.5 nanometers with a polydispersity index of 0.2. Less than 0.5 is already always acceptable. And the geotopotential of the nanoparticle I found as minus 8.96 millivolts. So I also checked its stability, and the stability after 6 months is 116.5 nanometers in size, and the geotopotential is also within the acceptable range. So later on, I checked its size with FESM studies, field emission scanning electron microscopic studies, and I found the average size by the histogram as 12.6 nanometers, and the A image will show how the nanoparticle will appear. And later I found that the presence of silver, I tried to find the presence of silver by energy dispersive X-ray EDX analysis. It shows the presence of silver at 3 kilo electron volts, indicate the presence of silver in allantoin silver nanoparticle. You can see the peak, the second peak, and the first peak, which is highest, is due to the background, silicon glass. And the next, I did the melting the temperature studies by differential scanning calorimetry and DSC. And I found that at first the peak was, the significant peak of the allantoin was found with distinctive endothermic peaks of allantoin at 237 and 256 degrees centigrade. So the allantoin peak was confirmed. And later I checked the allantoin AZNP peak and the peak temperature, which is called, at which the rate of heat flow into and out of the sample is at its maximum during the transition is called the peak temperature. The definition of the peak temperature is the rate of heat flow into and out of the sample is at its maximum. So the peak temperature is different for allantoin and allantoin AZNP, which shows that the allantoin AZNP formulation is different when compared to allantoin. And later, as the characterization was done, we checked the cytotoxicity studies, the safety studies on THP1 cell lines. And I found that when compared to the standard amphotericin B, the nanoparticle is 3.4 folds less cytotoxic and 10 folds less hemolytic. And I also checked, as the cytotoxicity and hemolysis are less, whether the uptake into the macrophage is is there any difference in the uptake by ICP-MS studies, inductive coupled plasmon mass spectroscopy studies. Through that, we can check how much metal entered into the cell. So by the ICP-MS study, I checked that the macrophage uptake is more in allantoin AZNP silver nanoparticle when compared to the normal citrate reduced silver nanoparticle. So even though the uptake is high, the toxicity is less. And later on, I confirmed the anti-leishmanial activity studies on the Leishmania parasite, which creates the leishmaniasis disease. And I found the IC50 on the promastigote form. Promastigote form is 8 microgram per ml, whereas on the amastigote form, which present inside the macrophages, I found it as 75 microgram per ml. IC50 means half of the 50% of the parasites were killed at which dose. So for amastigotes, I found 75 microgram per ml, and for promastigotes, I found 8 microgram per ml. And I also confirmed that by microscopic images, and you can see the morphology was changed in the allantoin AZNP, which becomes round and circled when compared to the control one and the allantoin one. Allantoin is not that much effective, and so there isn't any change in the morphology of the Leishmania cells. But we also confirmed it with FECM studies, field emission, scanning, electron, microscopic images. And the first A1 is the control, in which the flagella and the size of the leishmaniasis is normal. Whereas in the B, you can see that cytoreduced DGNP treatment, some of the cells, the star size, the star sign, which indicates that the cells are shrinked. In the B image, you can see that. And in the B image only, you can see the plus sign also, which shows that the cell bodies became rounded. And the arrowhead shows that the shortening of the flagella in the cytoreduced DGNP, but it is very less. When compared to the allantoid DGNP, you can see the flagella shortness and the shell shrinkage and the rounding was more when compared to the cytoreduced DGNP. It clearly says that the allantoin nanoparticle has less minicidal activity. So later on, as it was killing, by how it's killing to check its mechanism, and we studied the ROS, how much ROS it is producing, because for a majority of the metals, metal nanoparticles, the ROS is the major intermediate for the killing. So the ROS production is more in the allantoin AZNP treatment when compared to the allantoin treatment and cytoreduced AZNP treated. As the ROS is more, it will always activates the caspase pathway. It will always activates the caspase 9, and caspase 9 will activates the caspase 3 pathway. As the caspase 3 pathway is more, the caspase 3 activity will also be more. So we checked the caspase 3 activity by FACS, fluorescent assisted cell sorting instrument, by using anti-casp antibody, anti-casp3 antibody. We found that 4-folds more caspase 3 compared to the cytoreduced AZNP. Allantoin treated AZNP have 4-fold more caspase 3 compared to the cytoreduced AZNP. So as the ROS is more, the caspase 3 activity will also be more. And as the caspase 3 activity is more, caspases are nothing but those are the proteases. Caspases are nothing but those are the cysteine aspartate, cysteine dependent aspartate specific proteases. And also caspases will activate CADs, that is caspase dependent DNases. So the DNases are active, as the caspase activates DNases, the DNA will be fragmented. Nucleases are nothing but they cleave the nucleus, or the DNases, they cleave the DNase. So the DNase they will cleave, so we check for the DNA fragmentation. So in that DNA fragmentation by the FACS assay, we found that 2.75 folds more in allantoin AZNP compared to cytoreduced AZNP. The DNA fragmentation is also more. As the DNA fragmentation is more, we decided to check what cell cycle it is arresting. So interestingly, we found that allantoin AZNP arrests the cells at S phase 40.4% when compared to the control. It is 12.5%. So the S phase arrest is happening in the promastogotes. So later on, we need to check what leads to the S phase arrest, and then we found that as the ergosterol is the major pathway for the leishmaniasis, so we had first checked for ergosterol depletion assay, but we didn't found any depletion in the ergosterol content. So we go for the proteomic studies to find out which proteins are affected and metabolites are affected. So in the proteomic studies, we did 3 triplicates for the control and 3 triplicates for the treated. So to find out the after the LCMS studies, proteomics by liquid chromatography and mass spectrometer studies, we did the in silico studies by bioinformatics, MetaBioAnalyst software, and we found that the triplicates are similar. By the score plot, we found that the similarity in the triplicates which are acceptable. And later on, we found that upregulated and downregulated proteins in the heat map. Total, we found 6,303 proteins were detected, and among those, 205 proteins were downregulated, and 117 proteins were upregulated. And that is in the heat map, and in the fold change values also you can see in the B slide. The red ones are the upregulated proteins, and the blue ones are the downregulated proteins. And among the top 50 proteins, majority of the proteins are 60S ribosomal proteins. And you can see in the slide where I have isolated, those are the 60S ribosomal protein L13 putative, and RNA ribosomal protein L37, and RNA pseudouridylate synthase family proteins 1, and 60S ribosomal protein L37, And also the proteasome proteins are also affected more. So I did the gene ontology enrichment analysis by David software to check which biological process proteins are more affected. And I found that through the gene enrichment analysis that translational proteins in the biological process are more affected. And in the cellular process, ribosomal and cytoplasmic proteins are more affected. And in the molecular function, structural components of ribosome proteins are more affected. You can see by which I have highlighted with red marks, red arrows. So, and KEG, Kota Encyclopedia of Genes and Genomes, KEG pathway analysis. We can find out which pathway proteins are more affected, and in this also I found that ribosomal proteins are more affected, along with biosynthesis of ribosome proteins are more affected, along with biosynthesis of secondary metabolites pathways also. So later I did ribosomal proteins by string interactions to find out which proteins are more and which are closely interacted and related through the string assay. String is nothing but search tool for retrieval of the interacting genes, is a biological database which is online software. It predicts the protein-protein interactions. Majority of the proteins which are interacted are ribosomal proteins. You can see that in the left side. These are the ribosomal proteins are more interacted. And in the table also, I mentioned 60S ribosomal proteins, L37A and putative 60S ribosomal protein L13A and putative 60S ribosomal protein L2 and putative 60S ribosomal protein L31. The fold changes are decreased. So there, along with that, I also did some literature survey which shows that ribosomal proteins have a role in the DNA damage and apoptosis. And ribosomal proteins, this proteomic study shows the synthesis of ribosomal protein and protein biosynthesis machinery is downregulated, and downregulation of ribosomal proteins and elongation factors has earlier proven to be involved in the apoptosis. Later I did metabolomic studies also. Now it's clear that by the proteomics, majority of the ribosomal proteins are downregulated, which are involved in the translation process. Involved in the translation process. So I want to check metabolites of which metabolites are affected by the treatment with allantoin AZNP. So I found that by the LCMS only, I did proteomic studies, and total 3,845 proteins I found. And among those downregulated are 1215 proteins, and upregulated are 856. Yeah, I found metabolites 3845. And among those downregulated metabolites are 1215 and 856 metabolites are upregulated. And the A image is the heat map of the metabolites which are downregulated and upregulated in comparison with the normal site rate reduced AZNP. And the B side is the fold change values. The upregulated metabolites are on the right side and the downregulated metabolites are on the left side. Upregulated one is on the red color, which is on the downregulated one is on the blue color. And this study was also done using the Metaboanalyst software, and we found that majority of the metabolites which are affected were in the pathway of arginine proline metabolism pathway. The A image will show what is arginine proline metabolism pathway. This is the arginine proline metabolism pathway. In the arginine proline metabolism pathway, majority of the amino acids are downregulated, like ornithine, spermidine, L-proline, D-proline, and those are downregulated. Those are amino acids which are downregulated more. And so then I did the metabolomic set enrichment analysis to find out which process it is affecting more. And here also it is showing that arginine and proline metabolism pathway is affected more. And I also get the pathway analysis through Metaboanalyst. And I found that arginine and proline metabolism is affected more. And the pathway impact, you can see the x-axis is the pathway impact, which I put the arrow that was having the more impact with the minus log values more. If the minus log values are more, then it's having the less p values, which are significant. And these are some of the metabolites which are downregulated, indicated in the box plots. And this will lead to the apoptotic cell death, like ceramide and L-serine. And this paper supports that targeting the proline, glutamine, asparagine, arginine metabolomics axis in amino acid starvation cancer therapy. The amino acids are downregulating in my study, and the amino acid starvation will help in the cancer therapy. It was stated by the paper. So as a conclusion, for anti-Leishmanial study, allantoin shows the size of 128.5 nanometers and zero potential of minus 8.96, and with a conjugation of 42%, and it is less cytotoxic and hemolytic. Allantoin has anti-Leishmanial activity. For mashed goods, it is 75 microgram per ml IC50, and for pro-mashed goods, 8 microgram per ml. And ROS production is there. As the ROS production is more, it will enhance the caspase 3 activity. As the caspase 3 activity is more, it will enhance the CAD enzyme activity, which cleaves the DNases. So the DNase fragmentation is also more in apoptosis. And later, we checked the proteomic studies. In the proteomics, I found that the proteins of translational regulation proteins are more affected. And through the metabolomics, majority of the amino acids are more affected. I found that. And as there is a similarity between the cell wall nature of the anti-Leishmanias and the fungus, I also did additional studies on the fungus. Now we can see the antifungal studies. In the antifungus, we did the zone of inhibition on the saborodextrose broth agar, SDB agar. We did antifungal studies on Candida albicans treated with allantoin AZNP, and amphotericin B we took as a control. You can see that A image which shows the zone of inhibition of AMB, which is more, and allantoin AZNP 1x and 2x, it is a dose dependent. You can see that. And we also find out the IC50 as 8 microgram per ml. We also found out the IC50 as 10 microgram per ml. It's 8 in Leishmaniasis. In the B curve, you can see that viability assay we done on Candida albicans with the treatment of allantoin AZNP. So we also shows the antifungal activity on by the assay of colony forming units on Candida albicans after treatment with allantoin AZNP. And we found that 97% of the fungus is killed. You can see that differentiation. The second one, which is this is the second one, is of control. Here the number of colonies are more, and this is allantoin AZNP treated with dose. And we also checked the uptake of allantoin AZNP inside the Candida albicans cells by using the ICP-MS, inductive coupled plasma mass spectroscopy. And we found that the uptake of allantoin AZNP is more when compared to the characterized AZNP into the Candida albicans. As the uptake is more in the Candida albicans, we go for its mechanistic studies to check the ROS production. And interestingly, we found that the ROS production is more, as it is a metal nanoparticle. It's the most common killing effect. So the ROS production of allantoin AZNP is more than CAZNP in Candida albicans. And we also did the apoptosis and necrotic assay by using propionamide and enixin V. And we found that in the Leishmaniasis, we found the killing is through apoptotic pathway, but in the Candida albicans, we got the data as a necrotic pathway. Here you can see these are the 4 quadrants like Q1, Q2, Q3, Q4. Here the Q3 is AV negative and PI negative, which shows the live cells. And in the quadrant 4, which is A positive and PI negative, it is a early apoptotic sign. And the quadrant 2, it is late apoptotic sign. This is early apoptotic, and the Q1 is the necrotic sign. In the Q1, the PI positive cells are more, And in the allantoin AGNP treatment on Candida albicans, we got the PI positive cells are more and leads to the necrosis. You can clearly see that the cells are moving from Q3 to Q1 quadrants. In the allantoin AGNP treatment, these two are the allantoin AGNP treated. The cells is moving from the Q3 to Q1. Directly those are moving. But in the leishmaniasis, the cells are moving from Q3 to Q4 here, which is the apoptosis. So in the leishmaniasis, I got the apoptotic pathway, and in the fungus, Candida albicans, after treatment, I got it as a necrosis. So after this necrosis pathway of cell death, we are trying to check which cell cycle phase is arresting, and we found it as G0 by G1 cell cycle is arrested. You can see here in the allantoin AGNP treatment, the percentage of cells are more in this G0 by G1 phase when compared to the control one. It indicates that the cell cycle arrest is happening. And we also find out the negative. We also find out its antibacterial activity, which is negative for antibacterial. This states that the formulation is more specific to the fungus and parasite only. And we got very less antibacterial activity when compared to the cytoreduced AGNP. The conclusion of antifungal activity of allantoin AGNP is that it's having the IC50 of 10 microgram per ml, and it is showing the death through necrotic cell death, and it's through the ROS production. And this cell cycle arrest is happening at G0 by G1 phase. By using the allantoin AGNP, we can in the future we can formulate the topical formulation for skin infections caused by the fungus and leishmania majors like cutaneous leishmaniasis also. Since allantoin is the very safe drug, it is already used for cosmetic applications in the nano formulation also. The take home message of this presentation is that the planning is the crucial thing for a success. Success is the residue of perfect planning. If you plan it and you think you have an idea and you do it again and again, you will lead to the success. You need to change the plan if you fail it. And finally, I want to acknowledge my guide, Dr. Devabrata Mandal, and to the Heredity Biosciences for giving me this opportunity on this very important day, like Foundation Day. I'm happy to hear that. And congratulations to the Heredity Biosciences, and thanks to Ms. Sushri Swagatika Madam and Gopal Purohit sir, and to the funding agency like Ministry of Chemicals and Fertilizers, Department of Pharmaceuticals. Thank you. If there are any questions, I'm happy to answer it.
Thank you so much, sir.
Yes, sir.
Thank you, sir. So now the students, this is now for this written for the discussion. Any of these students have a question?
Yeah, before questioning, can I talk like right now I am in the Sri Vishnu College of Pharmacy and I'm working on nano formulations. I'm working on nano formulations against antimicrobial activities. In this college, I'm doing studies on even on the resistant strains also, resistant strains of S. aureus and E. coli. Along with the normal strains, I'm happy to collaborate with anyone if you wish to work with. Yeah, now welcome questions.
Thank you, sir.